Today, before I'm going into that sleep, but I little bit talk about the circadian clock, especially tissue clearing and also whole brain imaging of the every single cells. Okay. So a couple of years ago, 2014, we published two papers in Cells. One is for the adult mice brain tissue clearing and also imaging, and also the other one is application of this technique to the entire mice bodies. Okay. And then recently, we finally succeeded to apply this technique to the biological timing problem, circadian clock. So within our brain, like also mice brain, there is a tiny piece of tissues called suprachiasmatic nucleus, which is a kind of a center of your circadian clock. Okay. So during the day, neural activity is high, which is visualized by the C4 immunostaining. And then during the night, it's low. And then you can repeat, you can see the repeat of this activity over 2 days in constant darkness conditions. And as you can see here, we repeat this experiment, and then you can see the nice reproducibility of the C4 whole brain C4 immunohistochemistry, okay, which is already known in the slices. But here, what is different here is this is a whole brain imaging of the immunohistochemistry of the C4. Okay. When you apply this technique and then made an image, kind of a shoot movies, you can see the wave of the C4 activity. Okay. And then when we apply this technique to the entire brain analysis, you can see that almost half of the entire brain regions oscillate with different phase. So there are more than 640 different brain regions in Allen Brain Atlas. But half of them, or more than half of them, have a significant oscillation with different phase, peaking, for example, dawn or morning, or sometimes afternoons, or evening, or early night or late night. So what is interesting here is the cortex is peaking at the end of the active period, end of the night, but hippocampal region, which is related to learning and memory mostly, are peaking completely opposite to the cortex. There are 12-hour separation of temporal activity, so which is quite interesting. So we are now preparing this database, which may be hopefully available soon. So if you have your interest, the region of your interest, please check it out. Okay. And also, this kind of a technology, tissue clearing technology, is applicable not only to rodent, but also to the primate, even humans. Okay. So if you are interested in the first ever transparent human, if you want to be a first transparent human, please let me know. Okay. I cannot guarantee you are still alive after this, process. But I assure you will be in the, kind of history. Okay. Okay, so let me go back to the sleep. Okay. So we sleep scientists basically address two questions. What is the mechanism of sleep? First question. And then second question, what is the meaning or function of sleep? Okay. So let me first focus on the mechanism questions and then touch upon the function question later. Okay. So one of the fundamental sleep question, mechanistic question, is on the sleep called sleep homeostasis. So one night, if you cannot sleep well, next morning or next day, you feel sleepy. That's it. Okay. So and then basically neural activity usually counted and then memorized and then converted to the sleepiness. So what is interesting here is the timescale differences. Okay, so neural activity is usually millisecond to second, but sleepiness may last hours, even days. So there must be a molecular or cellular mechanism which memorize such kind of neural activity and then count it up and integrate and convert it to the sleepiness. But that kind of a molecular or cellular mechanism is totally unknown, at least in mammals. Okay. So today, because of the time, so I mainly focus on the one specific, kind of conclusions. So there are many ideas regarding the sleep homeostasis, but what I want to emphasize is you don't need sleep substances. Okay, you just need the wake-related substances like calcium, and then what you only need is memorizing of the activity or history of the calcium dynamics. Okay. So I'll tell you the kind of molecular evidence of it, but basically, so calcium is usually associated with the neural excitability, and then under the control of the calcium, there are a couple of very interesting enzymes. So calcium-dependent enzyme, including CAM kinase 2, and also calcinurins. So I will touch upon this topic. But before that, so I just want to introduce you to the basic, technological development. So to work on the sleep-wake cycle in mammal is very challenging, because if you want to test your hypothesis, so you need a lot of time. Usually, if you come up with the one great idea, it took 1 to 2 years to test. Okay? If you are lucky, you can get a very significant phenotype. But if you are not lucky, you will end up with a very beautiful negative result, right? After a couple of years. And so that is why we want to avoid such kind of situations by, shortening this kind of a process by avoiding the crossing or breeding. Okay. So let's imagine what if you can generate knockout mice or knock-in mice within one generation, meaning 3 months or 4 months. So we conceived this project idea called next generation genetics, defined by the genetics without crossing. Around 2010, and it took 6 to 7 years, so we succeeded to make a knockout mice by using triple CRISPR technique, which is already published, and also ES mice technique to make knock-in mice. Okay. So by combining these next generation genetics, next question is to perform the phenotyping. So for that purpose, so we try to avoid the invasive measurement, which usually required for the EEG and EMG recording in sleep. But to avoid that, we use respiration data, which is measured by the piezo sensor, and then monitor and also classify into the sleep or wake, non-invasive manner. So we design the LAC biocells, and then we design the chamber biocells, and then we design LAC biocells, and then we are lucky enough to get good support from the RIKEN, which is a governmental institute, to have more than 1,000 cases dedicated for the sleep-wake cycle measurement. So basically, we can measure more than 1,000 mice per week. Okay. So by combining these technique, so this is a kind of accidental finding, that we knocked out every single acetylcholine receptor, and then we found muscarinic receptor 1 and 3 have a huge sleep phenotype, reduction sleep phenotype. And then if you combine both, we found in this double knockout mice, impaired almost totally REM sleep. So there is no REM sleep at all in these mice. Okay. So meaning these genes, muscarinic receptor 1 and 3, together first essential gene reported in mice or any other species. Okay. After we discovered this technology, there are many newspapers covered because phenotype itself is shocking. And the UK newspaper is also report our finding, but the UK people is a little bit harsh. And then newspaper said, Japanese researcher disrupted dream. Okay, so how nice. Okay, so but it's well covered. Okay, And then after that, we also, continue this research. And then muscarinic receptor 1 and 3 is a Q couple GPCR. So obvious candidate underlying mechanism would be a PKC or IP3. So we focused on the PKC, which has 7 genes in mammalian genome, and then we knocked out them all. And we found one of the isoform, which is a PKC gamma, have a LEM reduction phenotype, reduced LEM phenotype. Actually, transition probability from non-LEM sleep to LEM is severely disturbed in these mice, which might be related to the LEM homeostasis. Okay. So that is unpublished data. Okay. But so by using this kind of technique, you can access to the very important phenotype information in vivo or in mice. Okay. So let me go back to the calcium story. Okay. So there are many calcium channel or pump in mice. Some of them are lethal, some of them are non-lethal. So for non-lethal gene, we can make every single knockout mice, and we found reduction of the calcium leading to the reduction of sleep, meaning usually wake-related substances somehow or indirectly might induce or related to sleep. Okay. And also, if you knocked out NMDA receptor, another voltage-gated calcium channel knockout, also leading to the reduction of sleep, okay, which is expected and consistent. And then again, calcium-dependent potassium channel knockout, also leading to the reduction of sleep. So calcium input pathway or excretion pathway might be positively involved in the induction or maintenance of sleep. And then on the other hand, calcium pump knockout leading to the significant increase of sleep. So magnitude is a little bit, small, but it's repeated and then confirmed. Okay. So calcium dynamics might be involved. So that is why we get interested in the downstream pathway, because calcium dynamics is so fast, which does not explain about the dynamics of the sleep homeostasis. So that is why we get interested in the enzyme underneath the calcium. So one of the obvious candidates was a CAM kinase 2. And then we made a knockout mice, alpha and beta, and we had a phenotype, meaning calcium-dependent enzyme like CAM kinase 2 positively involved in the induction of sleep or maintenance of sleep. So based on these results, we proposed 2016 April, so we proposed calcium-dependent phosphorylation pathway might be involved in the sleep homeostasis. We call this hypothesis as phosphorylation hypothesis. Okay. So after that, there are many another candidate comes in. For example, in autumn of 2016, another Japanese group reported SIK3 as a second sleep promoting kinase. And also Lausanne group also reported ARC1 and ARC2, which is a MAP kinase, also reported as a third sleep-promoting kinases. Okay. So, and then after that, there are many proteomics papers coming out in Science and Nature, and then reported there are many substrate candidates maybe explaining about the sleep homeostasis. So for us, CAM kinase 2 seems to be not only enzyme but also as a substrate. Okay. So meaning CAM kinase 2 might be a PEM, but also might act as a paper. Okay. So because CAM kinase 2 are well-investigated kinase in learning and memory context, activated by calcium and calmodulin, and then activated in the form of the autophosphorylation. So they get phosphorate itself. So meaning it acts as an enzyme as well as a substrate. Okay. And then activated CAM kinase 2 enhance the kinase activity, which is a kind of output of this kinase. Okay. So this is well investigated in learning and memory, but not in the sleep. So that is why we try to test many hypotheses by ourselves. And then one of the, so I only tell you the part of the, our data, but when you mutate to mimic autophosphorylation, you can induce the sleep enormously. And then if you inhibit this kinase by using the peptide inhibitor, you can reduce down to the 500 minutes per day. So you can almost double the sleep durations. Okay. So how about the quality? So regarding quality, so you, which is measured by the delta power of the EEG, so you can reduce the quality of sleep more than 40%. So and also you can increase delta power when you activate CAM kinase 2. So quality as well as the quantity are almost double by manipulating single class of the enzyme. Okay. And then interestingly, these kinases have a multiple phosphorylation site. So possible phosphorylation site is almost 69 in beta isoform, 54 for the alpha isoform. And then we introduced every single phosphomimetic mutation in the CAM kinase beta and alpha, and we found only one site significantly induced sleep. Okay? And then after that, we suspected, because sleep information may be important, so therefore it might be manipulated by the other phosphorylation. So that is why we try to screen the second phosphorylation site by additionally mutating the second phosphorylation site. And then we found second phosphorylation site at a different residue change the mode from sleep induction to sleep maintenance mode. And also third phosphorylation site abolish either sleep induction or sleep maintenance. Okay. This result is already published. I don't talk much today. But there are multiple status within the single class of the kinase. Okay. The next question is, what is the molecular mechanism? Okay, so in vivo and in vitro. So let me first focus on the in vitro system, which is pioneered by the Meditafsir group and Jim Kruger group, and then try to test the hypothesis calcium might be, significantly involved in the sleep, quality. Okay. So Meditafsir and Jim Kruger independently tried to culture the primary neuron for a couple of weeks, and then after that, they observed the nice synchronized oscillation, which looks like a sleep in vitro. And then when they administrated chemical cocktail, including dopamine, norepinephrine, histamine, acetylcholine together, they can induce the wake-like, behavior. Okay. So at that time, that was almost 10 years ago. At that time, I was a little bit skeptical. But after our in vivo data, which suggested CAM kinase 2 has a role, and also CAM kinase has a role in the cortical regions, so we start introducing this phenomenon system into our lab. Okay. And then we try to ask the role of the calcium first. Okay. So after we deprive the calcium by using the EGTA, which is a calcium chelator, and then original calcium concentration is about 1.8 or 2.0 millimolar. And then we added the similar amount of the EGTA to chelate the calcium. And as you can see here, you can see the increased firing rate, but also induced total desynchronization, which looks like a wake. Meaning calcium seems to be important for the, sleep-like, synchronization. But inhibition of the calcium may induce wake-like status. And then when we add back calcium, for example, 1 millimolar or 2 millimolar, you can induce slow wave-like oscillation, which looks like a non-REM sleep. Okay. And then here's the quantifications. So when we add back calcium, you can see the average firing rate is decreased because you can induce sleep-like, synchronizations. And then because of the existence of the downstate, average frequency is a bit decreased, okay, over the increased concentration of the calcium. And then which is evident by the power spectrum analysis. And then delta power means the slower component of the spectrum. Okay. and then you can see the increase of the delta power. Okay, but okay, so when you look at, so it looks like a, sleep status, at least in vitro, looks like an inactive state. But when you look at the data carefully, you notice very interesting things. For example, if you look at the maximum frequency of this state, you observe the increasing of the maximum frequency over the increase of the calcium concentrations. So meaning average frequency seems to be decreased, but if you look at the, maximum frequency, it looks like a sleep status might be a little bit active status. Okay? So please remember this, because I will touch upon this point when we discuss about the function of sleep. Okay. And then calcium might have a role in vitro, but how about the in vivo? Okay. To test this hypothesis, we try to look at the effect of the inhibition of calcium. Okay. So if the calcium only acts as a wake-related molecule, you may expect the suppression of the neural excitability when you inhibit the entry of the calcium. So if you believe calcium acts as a suppressor of the neural excitability by inducing sleep, inhibition of the calcium entry may end up with a hyperexcitation of the neural excitability. To test this hypothesis, we use a tissue clearing technique and then to look at the immediate origin. And in this case, not the C-force, but the ARC. And then we observe the hyperexcitation of the ARC genes all over the cortex. And then after that, we try to investigate the, identity of these activated neurons, and we found it not only excited neuron, but also inhibited neuron are excited. And then these neurons, almost all neurons express NMDA receptor, meaning implying that effect seems to be direct. Okay. So far, so we found calcium has a role, and also under the calcium-dependent enzyme like a CAM kinase 2 might has a role. And then within the CAM kinase 2, there are multiple status, including canceling status, meaning there might be an opposing kinase counteracting with the sleep-promoting kinase. And then to identify the wake-promoting kinase, we try to search for the different kinases. Okay. So one of the candidate was pKa, because pKa previously identified in fruit fly. Okay. And then we made a knockout mice, and then we found inhibitory component of the pKa induce reduction of sleep, and also reduction of the sleep quality. Okay. And then if you inhibit the pKa by using the inhibitor, you observe the induction of sleep, and also induction tendency, increasing tendency of the quality. Okay. So pKa might have a role as a wake-promoting kinase. And then if so, you may expect sleep-opposing phosphatase, so sleep-promoting phosphatase. So to test this hypothesis, we try to test every single phosphatase expressed in the brain, and then we perform the screening of the 34 genes. And then we found just one interesting phosphatase, which turned out to be PP1. Okay. And then this is a kind of a positive component of the PP1, which localize PP1 into the excitatory postsynapses. If you knocked up them, we observe the reduction of non-REM sleep. And then if you localize PP1 onto the excitatory postsynapses, we can induce sleep. But without localization, nothing happens, meaning the localization onto the excitatory synapses seems to be important. Okay. That is the story of the, non-lethal gene. But how about the lethal genes? So there are a couple of candidates, which include calcineurin. Okay. So that was a very difficult experiment, but we finally made a knockout mice by using the adeno-associated virus, which have guide RNA, and then inject them into the postnatally into the mice using the adeno-associated virus. Okay. By using this technique, we postnatally knocked out calcineurin, and we found reduction of non-REM sleep and the reduction of the delta power. And then if you activate calcineurin and then localize into the excitatory postsynapses, so we found the induction of non-REM sleep and also induction of the delta power. And then actually these two components have a kind of a competition in the excitatory synapses. Okay. So here, so we found wake-promoting kinase and also sleep-promoting phosphatase. Okay. So far, so good. But how about the function of sleep? Okay. So as I said, sleep are usually regarded as an inactive status. And the famous hypothesis called shy hypothesis basically said, during the wake, you learn something. During the sleep, you forget something. But as I said, sleep seems to be a very inactive status, okay, like this. Okay. To have a sleep, you need to be active. That's a counterintuitive, but the data suggested. Okay. Not only in vitro, but also in vivo data suggested. So maximum frequency is a bit higher during the non-REM sleep compared to the quiet wake. Okay. And if so, or if you, suppose such kind of firing patterns, and then you suppose Hebbian rule or STDP, which is already observed in the cortex, so we would like to ask theoretically or computationally what happens. Okay. So one of the talented grad students tried to ask theoretically, if you input the sleep-like patterns into the learning system, or wake-like pattern into the learning system, and then measure the theoretically synaptic efficacy or synaptic weight. And what we found is a complete opposite what we know or what we believed for a long time. So we observed during the non-REM sleep, like firing pattern, synaptic efficacy is increased, and then during the wake-like patterns, synaptic efficacy is decreased. So firing patterns, average firing rate doesn't matter. Maximum firing rate matters. Okay. And then if so, because that is a little bit different from what we believed for a long time, so we name it wake inhibition sleep enhancement mechanism. So we name it as WISE. Okay. So I don't know to name new hypothesis WISE is wise enough, but it's at least it's easy to remember. Okay. So summary. So during the wake, so we believe synaptic inhibition might happen, which may induce sleep, and then during the sleep, synaptic excitation might happen. So if Shy is right, LTP might induce sleep. If Y is right, LTD might sleep. And interestingly, calcineurin, which is a famous molecule to induce LTD, seems to be inducing slow wave oscillations. And the pKa suppress slow wave oscillation. Okay. Okay, so maybe I will skip this. But during the wake, your synapses may be fragmented, and then this fragmentation might be sensed by a lower calcium input, which activates calcineurin to induce slow wave oscillation, which is good for the restoration of the connectivity. Okay. So by that, I'd like to thank our collaborator for non-REM sleep and REM sleep. And also I'd like to thank the mice, a lot of mice. So almost 100 mice, knockout mice are made in this study. And also I'd like to thank you. So you somehow a little bit suppress your sleepiness during my talk. So thank you for your attention.